This last session we have some short talks. I'll try to keep it short, but I want to do one thing. I would like you to consider a completely different kind of system for some of the questions that you've been discussing at this meeting. And it is really going hard towards a reductionist approach. It's very much sort of an artificial life approach. Thinking about some of the complex problems that we all like to think about from a really, really simplistic perspective. And it's almost absurd how simple this is, which is, I think, part of the fun. Like any model system that we work with, we have the advantage of understanding something important about the system, and like every disadvantage, it may be way too simplistic to capture all of the magic and interesting things that we're interested in, but that is still to be determined. I will tell you a little bit about liquid droplets, and in two aspects on information and cognition. So here's the outline, and let's get my pointer here. So I'll give you a short introduction to active droplets to show you a bunch of movies, basically, what's happening there. And I will speak a little bit about minimal cognition and information in these systems. And I'm going to do that with going through a few steps. First, how do we animate matter, which is the basic principle here. Establishing an interaction between this droplet and its environment, and then exploring the behavioral variability of these systems. So it's basically you mix these chemicals together, you then observe what happens over time, you quantitate what's happening, that kind of thing. So I'll show you more about that. And then finally, this kind of behavior variability that we observe, how can we put something in that system to, say, make it be programmable, so have a switch that we can kind of manipulate as an experimentalist to understand something about how you can change the behavior of even these very, very simple systems. That's the overall goal here. So this is what I'm talking about. This is liquid-liquid system. This is macroscopic. You can see the size there. And you're looking at an oil droplet moving around in a pool of water. And as you know, probably from your kitchen, these two liquids don't mix, and they don't do this kind of stuff. So you have a liquid-liquid system that does not mix. And the reason that this one is going to be moving around for us is because I've manipulated the system by adding a lot of chemical potential to the system. So I'm adding chemistry. It's a spontaneous chemistry that breaks down, so all of the ingredients for this self-moving droplet are up here in this box. So the nitrobenzene is a non-reactive body of the thing. That's the oil stuff. Oleic anhydride is the chemical precursor that adds the fuel to the system. And that breaks down over time, forming more surfactant. So it forms a fatty acid called oleic acid. That's the chemical part of this system. So the point is, think of that red droplet there as a car, right? And if you don't put the right fuel in your car, it doesn't move. That's what's happening here. So the droplets in your kitchen don't have the right fuel, so they're not moving. If you put the right fuel in them, it's a possibility that those droplets also will move like this. So that's the sort of basis of the system here. You can see when that droplet moves, it leaves a chemical trail, right? Because I have a blue pH indicator in the system that is monitoring the pH. So as the pH goes down, it's making that sort of colorless trail. That means the system is producing, it has a chemical reaction that is producing a product, which is the fatty acid. It is also producing acids, which is changing the pH locally of the system. So it's making a kind of chemical wave. The reason that it's moving is because that chemical wave is changing the surface properties of that droplet in a way that it makes it unbalanced, so that the tension on one side of the droplet is different from the tension on the other side of the droplet. So it's basically riding a chemical wave. The chemical wave is made by the droplet itself. So I'll explain that a bit more in detail, but that's basically what's going on here. So these are some of the kinds of fun things that we're interested in exploring in my laboratory. Some of the sort of ways we could have embodiment, includes some metabolism in this case. As far as energy, we're putting a lot of chemical potential in these things. We really want them to move. It's not limited. We put a lot of energy in there, and we're looking at these processes of self-motion. This is kind of where the edge is for our research right now. So then there's a lot of other things we can explore with this. For example, inheritable information evolution. This could be very interesting, certainly possible, but that's at the edge of where our understanding is with these systems. So I'll speak mostly about the first three. Okay, so it's surfing a chemical wave that it produces itself. That's what I just said. So it has to be able to sense that chemical wave. This is how the chemical sensor works in the system, and there are different systems that we have developed, and they all have this similar principle. You have a liquid-liquid system. So let's see if my area is. So this is the droplet. This is the environment outside. Oops, it's not working too well. There we go. And the separation of the boundary between the liquid droplet and the liquid environment is mitigated by a surfactant that is in the system. This surfactant that's shown here is oleic acid. That's the product of the chemical reaction that I talked about. The fuel produces this thing. That self-organizes at the boundary to mitigate the tension, so the actual energy that is between these two immiscible fluids. That's the sort of interesting key part of this. And because we have a pH gradient, which I showed you in that blue pool as it swims around, there we go. We basically have this oleic acid in two states. One is a protonated state. This is for all the chemists out there. And the other side is the deprotonated state, which has this negative charge. So it has a binary state. It could toggle between these states based on the pH of the system. So when the pH is very high, very basic, it's typically ionized and you get this blue form there. And on the other side, when it's very acidic, you get this protonated form, which is the red form on the other side. And so it could go back and forth between those two based on the pH. Okay, so we could then measure what's the tension at that interface. So that's the graph on the bottom there, which shows what's the interfacial tension as you titrate the pH. So as you go from neutral pH out to basic pH, there's a huge surface tension differential between them. That's the wave that it's surfing. It wants to minimize its energy, so it's going to move where the energy is lowest, so higher pH. That's what's happening here. And all of those little fatty acids are flipping around between protonated and deprotonated states, depending on the pH, and then mitigating this whole system. So that's the sensor. Very simple stuff. What's the motor? It's fluid dynamics. For those of you who love fluid dynamics, Navier-Stokes equations, for example. What we have here, as I explained, a differential between the leading edge and the trailing edge of that droplet, which has different tension. When you have that in a liquid-liquid system with a boundary that is a surfactant that could move, which this one can, you can get a flow structure. And the flow structures we get are convective structures. I don't know if in the back you can see that, but from here we can see in that black and white image that you have convective cells that are flowing, right? So that's the motor of the system. You have a fluid dynamical motor coupled to the surface tension differential at the interface, coupled to the reaction rate of the fuel that's in the system. That's it. That's all you need to get this thing to go. More detail for those of you interested in this, it's a Marangoni-induced instability, which is why you get the fluid flow, and you have a low Reynolds number, so this is a swimmer. So this is going to be swimming through viscous water, and that's how the thing is going to move for us. Great. Let's see if I can advance. Yep. Okay, so this is another one of these. Let me skip. I'll come back to this because I'll just show you this. Okay, here's a different one. This is a droplet system that's even simpler than the previous one because it doesn't have any fuel on it. But I told you that you have a body. It's a liquid system. You have this reconfigurable surfactant at the interface, which is susceptible to rearrangement depending on the environmental conditions. So the droplet doesn't have to produce its own wave. We could put a wave in there. So that's what this is going to show, I think. Yeah, there's a droplet. And on the right side of that image, I'm going to put the gradient. There it goes. So now that's a gradient, but it's a chemical gradient of salt in this case, sodium chloride. And that's just diffusing slowly across this slide. It's a glass microscope slide. And whenever it gets over to the left side of that slide, that droplet can feel then the gradient, and then it moves directionally towards that. So this is showing that even a simple car, if you put it on a hill without any gas, it can go down the hill. That's what this is doing here. So that's what we're just making a little hill. That hill that we're making is this. So that delta differential for that particular experiment is shown here when we measure what's the surface tension along that droplet. As we change the salt concentration in sodium chloride on the x-axis, we can get that dynamic interaction. So that's all. It's surfing down a gradient now that we prepared. Once it gets to the end of that gradient, it stops. So when the car gets to the bottom of the hill, it stops. Okay, that's what's happening here. Okay. So because of that, you've seen the stick-to-stellium already. This is a nice video if it plays with the, there it goes. Oops. Oh, it's giving away the punchline. That's too bad. There it goes. So that's stick-to-stellium, right? This is a cyclic AMP gradient that is being distributed by an experimenter with a pipette tip, and you can see that there's a sensing of that cyclic AMP, and you have the cytoskeleton rearranging so it could go towards the source of that. So that's biological chemotaxis, and we've heard a lot about that. That's a very complicated process. This stuff I'm talking about is basically doing the same thing. I can make a nice road of chemicals there and have this thing follow that chemotactically. No proteins, no DNA, simple chemical system, but in a sense, like there's sort of a behavioral aspect of the system is very similar to what you find in biology, which we find is pretty cool. So then you could do fun tricks like, can it do this? There's our droplet, the pink one. The one on the lower edge is the source of salt. So that's the chemoattractant one. That just has embedded salt in it. And the idea is, well, can you task these things to solve two-dimensional mazes? You think it's going to do it, David, right? Of course. yeah. The rat wastes a lot of energy, doesn't it? Yeah. That's a good point, actually. That's a very good point. So, yes, of course it can solve the maze because to us it looks like a maze, right? But all this is, again, is like what Richard was saying. It's it's like a ball rolling down a hill. I like that because we can use that as a null hypothesis for what's happening here. Ball rolling down a hill has some interesting dynamics. We can measure it. And then we will say, yeah, that has nothing to do with cognition, whatever you're doing. Just go away. That has nothing to do with cognition. So that's, I think, a very good null hypothesis. Is this just a ball rolling down a hill, even though it looks like it's solving a maze? It's just doing, like I said, it's just following this, right? Energy minimization. So that's all it is a ball rolling down a hill. So what's different or what's interesting about these systems? They are simple. For this experiment where I said the droplet has its own wave, let's see if that's going to go, yeah. Droplet's surfing its own wave, so it has its own energy, right, that is supplying to the system, its own sensor, its own motor. It's moving, it's patterning the environment. So we say in this paper that we published a while, chemical basis for minimal cognition. I think that's why I was invited to this conference. It has sensory motor coupling. It's simple, but it's working. And the cool thing, one of the cool things about it is that it is avoiding its own eventual equilibrium. It's burning fuel. It's going to eventually burn itself out. This particular system, imagine you have a compartment that is burning fuel, and due to Le Chatelier's principle, it's going to accumulate waste products around itself, and it'll reach chemical equilibrium. But you can delay that by just moving away from your waste product. So that's what this is doing, and actually the blue field one shows that pretty well, if I can get that rolling again. So yeah, it's producing that chemical stuff, right? This is this acidic fatty acids. And when it produces it, it actually uses that as a mechanism to run away from it. So it's delaying its own equilibrium in that sense. And so that may be something you would believe when I tell you that, or we can discuss. pushing it a little bit further, is this simple chemical system that is on its way to equilibrium but has an emergent mechanism to avoid that, to delay that, is that self-preservation? Are we looking at self-preservation that is behind this very simple behavior that's happening here? That's just a question with a question mark there. But that's interesting for living systems, and I think for a lot of people here, the motivation here is very interesting. Okay, that's step one and step two, just to give you an idea of what are we animating, what are we playing with. So at this stage, we get a chemical recipe that we invent in the lab, and we put it in there, and then we just do like image analysis, what happens, how are they moving around. And one of the advantages of using these super artificial systems, which you can't do very well in biology, is we can just change one parameter like body size. So this is what's happening in this particular graph. 1 microliter, 20 microliters, 50 microliters, small, medium, large droplets. Single droplets, they are activated with the anhydride, so they have their own fuel, and then we just look at they trace their trajectories over time as they move. And as you can see with your eye, their movement is different based on what's their size, what's their volume, right? We did this many, many times. We put this through a self-organizing map program to pick out what's the pattern of behavior that we see here. And what we find is that there are 4 main modes of motion for these droplets of these 3 different sizes. Fluctuating, circular, directional, vibrating, and you can see the characteristic droplet shape, because these are soft things, right? This is soft matter. So when they move, they can change shape as well. It doesn't have to be spherical all the time, so they can deflect from that, and you can see the kind of deviations here and the variety. So we have 4 different modes of motion associated with 4 different types of shapes of these things when you look at single droplets. And the single droplets can then transition between these. So those are the 4 modes of motion and the transitions between those modes of motion. So sometimes, for example, the smallest droplets stay in the fluctuating mode for a long time. They can transition to directional and then back again, things like this. So you get an idea of the overall diversity of this. So that's from the discussions that we're having in this particular conference. I'm telling you that a very simple chemical droplet can have different modes of motion, at least. There's diversity with a single droplet, and it could switch between these modes of motion. That's number one. Okay, so there's some diversity. There's another thing that's happening here. When you put these droplets together, so 2 of these activated droplets, they do stuff like this. Yeah, they're doing something, right? Right. So there's a dance going on here. The presence of one active droplet is influencing the behavior of the other active droplet, and vice versa. They don't tend to touch or fuse together, but they do dance around each other. So you could just do the tracking of these, mutual tracking of 2 droplets in the same experiment. What's the mutual distance, which is shown here? Yep, that's your mutual distance. And you can plot this out. And at the very early stages of the experiment, we see that 2 droplets in the same experiment tend to be close to each other without touching each other, right? 2 single droplets in 2 separate dishes that we then overlay the plots. That's what you see on the right-hand column. So that's the control. And this deteriorates over time. As the droplet runs out of fuel, then this sort of behavior stops. But in the initial stages of the experiment, for a few minutes, you see this kind of chasing behavior of the droplets. So they actually have some sort of repartee between them. And if they can participate in this collective behavior, we can then overlay these 2 kind of analyses. So here are the 3 different sized droplets. Can they perform this kind of dance together? Yes, yes, and no. So we've reached a boundary of this kind of behavior just by changing one variable, which is size. So what I want to show here, which might be, and this is what we argue in the paper, and this is really minimal cognition that I'm talking about here. It seems like even a simple chemical drop that is animated can have different diverse behaviors of motion, and the diverse behaviors of motion can then allow it to be more influenced by its environment or not. Okay, so here's the null hypothesis again that gets us pretty much nowhere in this discussion, is a ball rolling down a hill, right? That's what we're looking at here, ball rolling down a hill. But what seems to be happening is it can switch a mode of motion such that it then becomes decoupled from the environment. So your ball rolls down the hill and then it just stops. And then it re-engages and then it keeps going. This dynamic coupling and decoupling from the environment, we are arguing in this paper, is a basis for minimal cognition. I just make very clear what we're talking about here, which is different from what I've heard other people talking about. This is what we're interested in here. So, how am I doing on time, sir?
You have no time.
I have no time.
Because there's lots of talk. So if you could finish quickly, that would be good.
Oh yeah, I could finish quickly. That was the main point. And I want to just say, let's just do this one. This is a fun one. Here's a 2-droplet experiment that's different than before. We have a source and a sink droplet. So the little one is the source, the big one is the sink, and there is actual material transfer between the source and the sink. Okay, that's happening over a course of minutes and hours. You're looking at 3 different experiments here where we've changed the concentration of the blue dye in the little one, in the source droplet. And just by doing that, we start to see diversity in behavior from this sort of binary star motion, oscillating star chasing motion, just by changing, again, the concentration of one dye in the experiment. So that's the little dial I was talking about. I wish I had more time to get into this, but this has to do with this argument of where's the information in your system. Of course, we make the information by putting a 50 microliter droplet, 9 centimeter dish. We're doing that. But what's the thing in the droplet that we can pattern and program in that's information? What I'm showing here in a bunch of other studies is that you can just use some dyes. That's good, because then you know what the information is, because you put it in there and you can monitor it just with a camera. What's the color? What's the pixel density? Things like this, pixel intensity. And you could understand what the information content of your system is. And that then changes the behavior of the system. So that's my conclusion.
Awesome.
Thank you.
Okay. Yes, let's thank our speaker real quick. We have time for one very quick question while the next speaker, please, would come up. Gaspar. Oh, yeah.
Yeah, I can hear you. Yeah, I can hear you. It's fine.
I think the implicit assumption, or maybe explicit metaphor here, is likening these droplets to cells. But I was wondering if it's possible to make an analogy to, let's say, liquid-liquid phase separation inside the cell, RNA condensates and things like that.
Sure.
Is there anything about the physics of that scale that would that would allow this stuff to apply to RNA condensates?
It's a great question, and it has significant biological relevance, what you're talking about. I don't see it. I don't see it right now, because you do have a 2-phase system. But I'm wondering if your RNA protein aggregate inside of the cell is more like a coacervate, which is not necessarily like a liquid droplet system we're talking about here. But there are some interesting things we should discuss. Yeah.
Okay.
Thank you.
So let me just say something. So I totally miscalculated the time. And so this session is supposed to end at 6, and there's no dinner. So would people be opposed to ending at 6:15 or 6:20, and that would give everybody 25, you would give everybody the same.
Oh, sorry, I was just following the...
I know you were
following orders,
yeah.
I was just following
It's
orders.
what happens when you follow a bad leader, you get into trouble. Is that okay for everybody? Because, yeah, okay. So I think what we're going to do is...
Now we have more time for questions.
Yeah.
Yes.
Yes.
Please.
I'm going to let everybody, I'd like everybody to have a half hour or at least 25 minutes, for
You got
sure.
it.
Thank you.
Yeah.
You got
Thank
it. Got a question from the chat.
Question in the chat.
Thank you.
How does the variation compare when you test 3 droplets of the same size, say 5 microliters, with the droplets of 3 different sizes? Sorry.
Yeah.
Yes, does that make sense?
Yes, I do. Yes, I understand. So yes, that matters. We didn't do exactly that experiment, But as you can imagine, just from the first principles that I laid out here, that all of this stuff is going to matter to the system. Yeah. So it's going to be number, size of the droplets, and if I was still connected, I could show you. I could show you a video on that. The current experiments that will be reported, if any of you go to the Artificial Life Conference in Tokyo, in Japan in July, my colleagues will talk about this. We've gone up to 50 droplets and look at the group dynamics, and they're fascinating. And I wish I could show you the video of that. Because it's odd, not really intuitive. But they form very large aggregate structures that are dynamic, that collapse and reform over time. So they form, I'll just tell you, they form a boundary structure that's a ring. So imagine 50 droplets in a ring, right? They just self-assemble to form this ring, and then it breaks, and it crumbles down, and then it reforms. And then it crumbles down, and it reforms over and over and over again over the course of hours. So we see this very dramatic population level behavior that we don't yet understand, actually. Because, look, I told you this stuff is very simple, but to understand exactly what's going on is not so easy. Yeah, so that's the current thing.
All right, one last really quick question with a really quick answer.
Yeah, so sorry, thank you, great talk. So I study lipid droplets in vivo in terms of like triglyceride lipid droplets, and one of the main constituents of the monolayer would be phosphatidylethylamine. And it reminded me a lot of your sensor because it's Zwitter ionic, but upon a slight increase in pH, you get a negative charge, which is, some people have speculated may be important for the proteins being recruited to the droplets and the dynamic of the droplets. So I'm wondering if you tried to maybe make a more physiological one using a monolayer of phospholipids.
Yeah, that's cool because that actually supports when you're using your muscles, right? You're lowering the pH, and that should break down your oil reserves so you can use that as energy. So that makes sense physiologically, right? No, we haven't tried anything more complex than a single chain surfactant, but that could be done. Thank you. Yeah.
Awesome. So dope. All right, let's move on. Let's thank our speaker again.